We are live.
Hello everyone. Welcome back to the afternoon session on Midchem Webinar Series 2.
Department of Pharmaceutical Chemistry and ALUMNI, RBVRR Women's College of Pharmacy. It's my privilege to invite Dr. M. Sumakant Garu, Principal and Convener of this webinar, to speak few words. Welcome, ma'am.
Hello.
Welcome, ma'am.
Yeah. Hello.
Ma'am, we request you to give the welcome note.
Okay, but I didn't hear your voice. Okay, thank you. Good afternoon to one and all. I am Professor Sumakant, Principal, RBVRR Women's College of Pharmacy. I feel proud that to invite today's international speaker, Dr. Jennifer Mudunuru, and she is the alumni member of RBVRR Women's College of Pharmacy. And I also invite all the participants, colleagues, and dear students for today's session, that is webinar series 2, afternoon session. As I told before, at the time of the first series, the purpose of conducting this webinar is to aware all the professionals what are the computational techniques available to speed up their innovation process in drug design. In the first series, we were aware of the different techniques and basic introduction about computational techniques in drug design. In continuation of that, today we are at the afternoon session. Today morning also, she explained, Dr. Shirisha, the basics as well as the applications of these computational techniques in drug design, what she is doing in her lab with the students. I think with that, all the professionals were inspired and interested in doing the PhD, especially in pharmaceutical chemistry, to develop the new compounds and to make them as a drugs. Now, with that, in continuation of the morning session, in this afternoon session, it was started with Jennifer. She is the student of this college, second batch. She completed her B Pharm in 2011, and after that, she went to US to pursue masters as well as to complete her PhD also. So, as a principal, I am glad to see her today to give the talk on this computational outlook in drug design. Now, I think decade by decade, the advancement in computational outlook we observing, especially with artificial intelligence and machine learning techniques, with this big data, there is a lot of changes in drug discovery also. How it, at the beginning, it is random screening, later on target based, now it is personalized drug development. So why I'm telling this personalized drug development, whatever the data which is available now with diseases as well as the targets environment, now we are in a position to develop the drugs which is suitable for a person. So with the group of persons, whatever we are getting the data, so based on the data and by changing the existing drugs, today we are saving lot of money as well as the time. Now we are able to develop the new drugs. So for this, what internationally the computational techniques are available, will come to know through this afternoon talk by Dr. Jennifer. So once again, I am inviting Jennifer to continue the talk.
Thank you,
Thank you.
ma'am. Can I sound check? Can you guys hear me okay?
Yeah, it's okay.
Okay.
Clear.
And can you see the presentation?
No.
Can you let me know when you can see the presentation then?
Yeah.
Okay.
Try it.
All right.
Not visible.
So screen share. Okay, so it says it's sharing. Can you confirm if you can
Ma'am,
see
it's
the
Not
screen?
yet.
ma'am, it's entire screen.
No.
Not the presentation. We can see.
So can you see the presentation now?
Yes,
No,
please
ma.
make it
Okay.
full screen, ma'am. Ma'am, make it full screen. Yes, perfect, ma'am.
Yeah.
Okay. Sounds good.
So
So thank you for the wonderful information and introduction, Sumakant ma'am. I'm really honored to be here today. It's a huge thing coming back to the place where it all started to give a talk. It's more like sharing my experience and what I do, how the journey was, and what I do currently right now. So, like ma'am said, I'm going to switch gears a little bit from pharmaceutical chemistry and drug designing to more of a PBPK modeling in discovery and development. PBPK is physiological based pharmacokinetic modeling. So, if this is the first time you're hearing it, you'll understand more as I go on. Um, I first wanted to thank Vijay Bhargavi ma'am and Sumakant ma'am for actually finding me and inviting me to give this talk. It's really exciting to come back where it all started to share experiences that I've learned and the knowledge that I've gained over the years in my journey here. So, with that, I just wanted to give an overview of what I did, where I did. I'm not going to spend too much time on this, but it's more like a snapshot of my journey. I was from the second batch of pharma students in RBVRR Women's College of Pharmacy. I graduated in 2011, came to US, I pursued my master's. I fell in love with pharmacokinetics. So that's what was my focus for the master's. I did it in Long Island University. Did a company-based project with isolated perfused kidney models, where you separate the kidneys from the animals, you run it in an in vitro system, and you get the data. So, with that love for pharmacokinetics, I continued pursuing my PhD in the same place with a grant from Onconova Therapeutics. It's a small pharma company. um, the milestone for that is when I finished my PhD, the data was used to file their IND paperwork for efficacy and talks, and they got really good feedback from that. That program is now in first time in human going on. So that's a huge moment of pride for me, being able to work on that. Um, Simultaneously, while I was doing my PhD, I was also working at a cancer research center on a research fellowship for 1 year, learning more about cancer cells and xenograft models, which means you induce cancer into small animals and then you work on the animals for experimentation to find if there is efficacy and safety. So it was a long journey. It was a journey of about 7 years or 8 years, and then I started working for GlaxoSmithKline, which is one of the large pharma here, from 2018, right after I graduated, and that is where I am right now. Um, Focusing on pharmacokinetics, but very special focus on DMPK and computational modeling. Um, so, to give a perspective, because when you're sitting in a bachelor's or a master's program, to give a perspective on where I sit in the whole pharmaceutical sciences spectrum, we know we've seen quite a few different specializations. So I went from there to pharmaceutics, and then from there into pharmacokinetics. So the department that I sit in right now, it's called DMPK, which is drug metabolism and pharmacokinetics. It's very focused on the pharmacokinetics and the metabolism of a compound, and it's PK. And my specialty is in modeling. So I used computational tools to gather all the data that is existing from in vitro, in vivo data, and to kind of support the process of drug discovery and development. So you'll see in more detail how it goes structure by structure. Um, so going to the next slide, I'm sure you've read this right from your bachelor's levels. I remember reading Subramaniam cover to cover like a multiple million times. So this forms the basis for what I do. So pharmacokinetics, I just wanted to touch what it meant. It's the disposition of the drug in the body. So basically, what the body does to a drug when you take it is pharmacokinetics. It's divided into four parts, which is absorption, distribution, metabolism, and excretion. So the snapshot here, the picture at the bottom, I just wanted to show that there is no limitation on what or how we can compute these models. It can be done for almost most of the available formulations and routes of administration, because we have oral, we have IV, syrups, capsules, tablets, etc. So there is like a whole bunch of things that could be done. um, so this picture on the right side, it's a snapshot of what all processes we study or are important for pharmacokinetics. So if you take a drug orally or any other route of administration, oral route is the more complex one, because then you have absorption, enterohepatic circulation, which means it goes into the gallbladder and then comes back to the gut. Then the drug gets metabolized in liver, and then it goes out. If it doesn't get metabolized, it becomes available in your blood, and then it goes to your site of action and to the various tissues, which is then called the distribution. So this is kind of leading you into the presentation, so I don't start saying things that are relatively very new and unfamiliar to you. So this is the whole process, when what happens when you take a drug into your body, from the point it enters to the point where it goes out of the body. It undergoes these four mechanisms predominantly. So that is called the ADME parameters, which is kind of the backbone of the pharmacokinetics, and applies for most of the formulations. So I know since I'm not the first one in the series of the webinars, and I've listened to the previous webinars, and they sit relatively in different spectrum of the overall drug development process. So this is to give you a snapshot of where we come in as DMPK scientists or pharmacokineticists. So you have disease related genomics, where you're trying to understand your disease, whether it's oncology or any therapeutic area. Then you target identify and validate, and you see if it's a good target and if you could develop any medicines for it based on structure. So once you have the target, then you go into the lead discovery and optimization. So you choose a target, then they make hundreds and hundreds of compounds that would actually engage that target. And there involves a lot of in vitro experiments at this stage, where you're trying to filter out all the compounds that are actually engaging with your target. And then you arrive at your drug candidates. It's never usually one candidate, it's normally hundreds of candidates, and all of them go through the screening process using in vitro systems, which is outside the body. In vivo is inside the body. So in vitro system is growing cells in a petri dish, doing work, adding the drug to it, looking if you have any target engagement, measuring the efficacy concentrations and things like that. So once you have screened the drug candidates, they now narrow it down to a few tens of compounds, or maybe I would say less than 10 compounds, which will then go into preclinical studies. Preclinical studies usually involve species like mouse, rat, which are the bottom of the spectrum, and then you go up into dogs, and sometimes they also do cyno, which is monkey, depending on where the target is. So we try to choose the species which are relevant to the targets that we are engaging. So we want at least some kind of similarity to the human in terms of target engagement, because if your targets are really different across species, then you might actually not see a translation from the preclinical species to the clinical species in human when you finally do it. So it's really important space where you're trying to understand the efficacy and safety of the compound using these preclinical species, um, but also maintain a translation into human. So it's really important that if you do it in rat, but if it doesn't translate into humans eventually, there is a gap, there is a knowledge gap. So it's a really interesting space. There's lots of opportunities there for drug discovery and optimization. Um, and then it goes into clinical trials. So this is where the physiologically based pharmacokinetic modeling actually comes into picture. It's not only in humans, there's also computational softwares where you can use the preclinical models. You build preclinical models in animals, which is a virtual model without using the actual animal, but you can actually create an animal physiology in the computer, and then you run predictions to see what might be the pharmacokinetics of the compound, or what is the toxicity levels, or what is the dose, depending on what the application is for that. So with that understanding, they then move on into clinical trials where we support a majority of applications in humans, starting right from the first time in human phase one. So if you are familiar with the development process, it's usually divided into phases. So it's preclinical, phase one, phase two, phase three, and then post marketing surveillance, or phase four. So this step is preclinical, and we get involved right from the preclinical stage whenever PK comes into picture. So with that background, if you move into the next slide, I just wanted to touch on what PBPK modeling is, if you have not heard that before. Um, and it's okay, I didn't hear about it until I was in PhD. It's a very focused and very fastly evolving science. And so I just wanted to touch on what it means and why use it. So physiological based pharmacokinetic modeling, also known as PBPK, it's a computer modeling approach which incorporates the various compositions in the body, like blood flow, tissue composition, organs, and everything, to define the PK of the drugs. So it was initially described in 1937. It involves a series of differential equations and calculus. If you're thinking where all of that makes sense in connecting the dots, it actually does. So everything you study eventually connects together when you are applying it to drug development. So all those differential equations and everything go in the background of such kind of computational modeling. So it's a powerful modern tool. So it's intense, it's complicated. It's a combination of mathematical equations to make quantitative PK estimations and predictions. So it's quantitative, it's not just qualitative, it's quantitative, which means that when you actually run a simulation, you're getting the real concentrations in your plasma, which is called quantitative PK. And it tells you with this model that you have built, this is the probable concentrations that you would see for this compound if you dose it at a certain dose in certain species. So there has been a renewed search. So when PBPK started off initially, which is probably about 15, 20 years ago, um, it was highly an academic exercise. So there were a lot of universities all over the world that were actually trying to use these computational methods, and it stayed academic for a while. And then there was this increased interest in the pharmaceutical companies and also the regulatory agencies. So whenever the regulatory agencies are really interested in something, the pharma industries try to keep up with the guidance and try to use that as part of their development process. So this is an excerpt from a paper from 2011, Zoe et al. There's very nice introductory papers on PBPK modeling that I can share um, at the end of the presentation for someone who's interested in resources. But he said that during the regulatory review of clinical pharmacology data in new drug applications and biologics, questions are routinely asked about how the intrinsic factors and the extrinsic factors might influence dose response and exposure response, and also the impact of these various intrinsic and extrinsic factors on the efficacy and safety of the compound. So, when the regulators started asking these questions, the one way to answer them was to use these computational tools. So, then the PBPK modeling became one of those tools that could be used to answer these critical questions and in a limited amount of time. So, time is of great essence when you're developing a compound. Right now, according to the 2020 um, numbers, it takes somewhere about 8 to 10 years to bring a compound from the very early stages into the market, and an investment of around $2.9 billion. Um, so you can see why time is of essence, because the faster you go, the faster you put the drug outside, but you want to make sure you're safe and your efficacy targets are reached. So that's why computational modeling has picked up such great importance in the last decade or so, with all the regulatory agencies like FDA is Food and Drug Administration for US, and then there's EMA, which is European Medicines Agency. They have all started um, asking for more PBPK as time goes by, and there is a lot of interest. So this has been a platform that was used quite a bit in recent times. So, what if you decide to build a model? What is the scheme? How do you go from box one to box four? So, as you see, there's preclinical phase one, phase two, phase three, then there's review, and then you have phase four, and the drug gets approved, and you have a post marketing surveillance. So, at these various stages, there's different spaces at which you can use the PBPK. So the first step is, once you have the preclinical data, like I said, that's where we most probably get involved, is you need to have some kind of input data that comes from the preclinical species. So what we do is we establish a preclinical model based on the available discovery and the preliminary in vitro data at that point in the development, and then we evaluate and expand the understanding to the compounds. So, for example, let's say you have 5 compounds and we have to prioritize which one will go further into development. And we have preclinical data from those compounds. We build a model, and then we run certain efficacy study predictions and safety predictions and see which one performs better. So it helps you prioritize within those compounds. And most probably at this stage, the data that is coming from is either from the animal species or from the in vitro data. So then we use all of that understanding to guide the first time in human milestone, which is the first time any human subject gets dosed. And it's an important one because you want to get the dose right. You don't want to have any toxic effects, but you also want to have a minimum amount of exposure so you see a effect for what you're trying to see. So with this preliminary model, we guide those human dose predictions for the first time in human studies. So once the first time in human studies, human trials are done, you get the very first PK data from the actual humans. Sometimes it is similar to what you have predicted. Sometimes there might be a knowledge gap and you might have under predicted or over predicted than what you actually see in the human based on the very first model. So then we take the learnings from this first time in human study and then we refine the model. So, as and when data keeps coming in, we keep refining the model and updating the model to a better place because now your data is coming from humans, which is the final species that you want to study in. So as the phases go by, as you go into phase two, you get more data like the ADME study, which is a very definitive study. So if you remember the snapshot a couple of slides ago, you get almost most of the information that has to do with metabolism or elimination, what organs is it going to, how much tissue distribution there is. All this information comes in at this stage. So we use all of that data and also definitive in vitro data at this point, and we update the model. And then we move into the next step. So as you go on moving into the next phases, the questions become very specific. So, for example, one of the major questions that we get is drug drug interactions. So, this is something that the PBPK modeling is used quite a lot because once you have a drug, you have dosed it to the human subjects. But if the human subjects are also taking some other drugs because of their coexisting conditions, then you would want to know if your new drug is going to affect the pharmacokinetics of those drugs. So it's called drug drug interaction. It can happen that one drug affects the PK of the other drug. It can either increase it and cause toxicity, or it can decrease it and cause loss of efficacy, because if you lost the drug, you will not see the effect. So if you're taking a pill for blood pressure, let's say, and you signed up for a clinical trial, if you're taking a new drug which is subliming the effect of your blood pressure drug, it's not good for the patient. So all these understandings come from the drug drug interaction trials. And that is one of the major things that we do using PBPK models. Even before going into the drug drug interaction clinical trials, you can first run those using the PBPK models. And not just drug drug interactions, they have expanded the PBPK modeling to quite a bit these days. You can check in various disease populations like renal impairment, hepatic impairment, cancer, quite a few actually, pediatrics. I have a slide to show you where all um, it can be applied. So if you build one model and verify it or upgrade it with your clinical data, and if you have enough confidence in the model based on the regulatory guidance, the applications of the model are way too many, and that's a good thing. So what platforms do we use? So I've spoken all about the PBPK modeling, and I know the first question I get asked is if this is an open source software. Unfortunately, they are not. At least these two that are predominantly used are not open source. But they are something that almost all of pharma has licenses to. They pay quite a bit of money to obtain these licenses just because of the importance of these platforms in the drug development phase in the companies. So the one that I am predominantly working on is called SimSip. Certara is the company that makes it. They also make Phoenix Vinonlin. If you have heard of that before, it is used to do NCAs and POP PK modeling. But they make SimSip, which is the actual software platform that we use to do this kind of modeling. The other competitor is Simulations Plus, that's the company, and they make a software that's called GastroPlus. SimSip and GastroPlus are similar on a lot of accounts, but they also have their pros and cons or advantages over each other. So today I'll show you at the end of this presentation a snapshot of how SimSip looks, what goes into the platform, and kind of get a familiarity to you guys, how everything goes into the system before we do the computational part of the modeling. So this was the slide where I was talking about is a PBPK model. If you look at the center part, a PBPK model has quite a few compartments. It takes into account the physiology, because it's a physiology based pharmacokinetic modeling, right? So it means that it's based on the physiology, which includes all the organs in our body and your venous blood and your arterial blood. So it's a complex model that actually takes into account your organ size, what is the blood flow to these organs, and quite a few parameters. So if you are able to build one model with enough verification and confidence from your clinical data, from the very first clinical data, the applications are very diverse. So you can use it for drug interactions, like we spoke. You can check if there is food effect. So for some of the compounds, if you take the medicine with the food, it has a reduced effect. For some compounds, there is an increased effect, but you won't know it until you go into the clinic and you get the PK data and you think, oh, I should have dosed it in fasted state because it's more effective. So, in order to save that time and kind of guide you whether you have to go in a fasted state or a fed state, which is called a prandial state, um, you can apply this model and run simulations and get that understanding beforehand, so you can save a lot of time and money. So there's also opportunities to do racial differences. You can There's quite a few populations that were built. You can do Japanese population, Chinese population, Euro-Caucasian population. The reason being, all of us are not similar in terms of our physiology. So your enzymes are different. The way that you metabolize a drug is found to be different in different kinds of races and ethnicity. So it becomes important, if you're making a drug for for a certain population, if you see a certain disease in a specific population, then you want to understand how it is different in those particular population compared to the other ones. So you can use the model in such aspects as well. And then there's hepatic and renal impairment and reduced cardiac output populations. What happens is these patients are often they often have very complicated pharmacokinetics because they have an organ dysfunction. And before you can go into a clinical trial in these populations, it's always advantageous to actually understand what might happen in those populations when you give your drug by using a PBPK model, so that your risk comes down significantly and you have a greater understanding and not blinded in your clinical trial with very adverse effects or toxic events or loss of efficacy, or that you just, it just went out of the system. It was never in the body because it was completely removed for whatever reasons. The other extended applications are, there's a cancer population that we can use, and we have quite found that it sometimes it's quite significant difference in pharmacokinetics because of various reasons how the physiology is affected in cancer patients. And the other one that is picking up really right now, because FDA and EMA have also started accepting models in that space, are the pediatrics, pregnancy, and preterm and neonates. So we often see clinical trials are in in the older adults. There's not a lot of clinical trials that go into pediatrics, but most of these drugs may also be used in a pediatric population if they have that particular disease state. But they are not actually studied in the pediatric population before they get an approval for many compounds. So now what they're doing is they are asking us to build pediatric model. You build an adult model and you can actually translate it into a pediatric model, and you can run simulations by dose adjustments and see what kind of exposure you might see in pediatrics or preterm populations or neonates, as well as pregnancy. Most of the clinical trials, in the inclusion exclusion criteria, they normally don't include the pregnant population because of the effects, and you don't know how the placenta will transfer the drug. It might affect the fetus. So it's a population that you don't normally recruit in a clinical study. So it becomes even so important to use a model to kind of target the pregnancy population, because sometimes you would have to use these drugs in a pregnant woman, and it's always advantageous to know how what kind of PK you might expect going into this kind of population. And in those instances, the PBPK model comes in very handy. So these are all the various kinds of applications that you can do with a single PBPK model if you build it from scratch up, from the preclinical, and then optimize it with your clinical data. So this is to show you a snapshot of how there was this surge in use of PBPK modeling across various therapeutic areas. So this is until 2018, and I believe this is over a period of 10 years. So 2008 to 2018, if I'm correct. And these are the various publications and applications of PBPK models that went into the various applications. So as you can see, there's quite a bit of DDI that we do, and then the PK special populations and other applications. So it's quite a bit, and it's not focused on one therapeutic area. It's predominantly used in oncology, infectious diseases, and cardiovascular, where you have high risk population and you want to understand more before you go into the actual clinical trial. So but you see, it's not restricted to those. You have quite a few gastrointestinal, immune suppressants, respiratory, and even supplements. It's coming up these days, and also biologics. So they have been expanding pretty fast, and it's even a developing. It's a very developing area in DMPK, which is really hot right now. So there's a lot of interest, and various universities are also coming up with programs that with our special focus in PBPK modeling masters. When I came here, there was no masters in PBPK modeling. It was more like a side gig that you learn by going to workshops. But now there's like very focused programs that are actually teaching PBPK modeling and computational modeling as the specialization in PhD and masters. So it's a similar representation. This is basically FDA approved products. So they've people have started taking approved products, and also it's not just already approved, but um, how FDA has accepted PBPK models in various disease areas. So there's quite a lot of acceptance these days. Previously, they were not very confident, probably 15 years ago, where it was still tested. They were not, but now FDA, EMEA, Japan, almost all of the regulatory agencies are asking for PBPK models, and they have more confidence in it, and they are building over the time, and they are also publishing guidances. FDA came out in 2019, I believe, with a PBPK guidance. So if someone's interested in this area, you can always look it up. And it is a really good guidance which tells you if you want to use PBPK models as part of your regulatory package, what do you have to do, and what all FDA expects you to do, and what are the norms and guidelines for that. So it's a really good piece of document. So now, with all that background, um, if we go into the next part, what goes into building a PBPK model, right? So there's two parts to it. There's intrinsic, extrinsic factors, and then the PBPK model components. I'll go into a little bit of detail in the next slide, but this is essentially to show that when you're building a PBPK model, it's not just the drug properties. A drug has certain properties, right? You have log P's and pKa's, so which is a very which is very specific to the drug compound. And then in PBPK modeling, you also have the physiology part, which has to do with the individual and not the drug. So your organs, your various organs and kidney, liver, how big they are, how's your blood flow, and all of that. So the intrinsic extrinsic factors are more outside. So if I'm a person that's taking the drug, there's other factors that determine how the PK of the drug will be. Like if I smoke, if I use alcohol, if based on the environment, is it a stressful environment? Sometimes stress changes your PK too. And what other drugs do I take along with this particular drug? So there's quite a few factors that play a role in how a drug goes through your system. And a PBPK model actually combines all these factors. It also combines the intrinsic factors such as your age, gender, genetics, and if you have any specific mutations or alleles in your enzymes, all those sorts of things. So it's very personalized. And the other part that is really, really helpful, if you look at the bottom right snapshot, is to show that when you run a simulation, you essentially are creating a virtual population. So it's like you're running a clinical trial in your computer. So you create different individuals of different sizes, different heights, different color, different gender, and you create that variability within your population. And the advantage is you can create two individuals, or you can create 200 individuals, or you can create 2000 individuals. So if you're familiar with statistics, you need a certain power to the number for it to be a significant number, right? So we normally create more than 100 or 200 individuals so that you can also look at the variability within that population. So that's one of the advantages, is you're not just creating one profile and saying, well, this one profile is representation of an entire population. You're actually creating a whole population and looking at various subjects, which would be a real case scenario when you go out into the clinic. You will have subjects of all sizes and forms and heights and weights. So that's one of the big advantages of using this PBPK modeling to create a virtual clinical trial population. I think I only have a couple more slides before we go into the demo and you can see the fun part. This is just a um, more detailed version of what goes in. We have already talked about this part, which is drug data, which is the compound specific parameters. You have systems data, which is a person or an individual specific parameters, and then the trial design. So it's also an important part of the overall PBPK building process, which is how much dose do you want to give? What is the administration route? What are the other drugs? What population do you want to use? So this is all the study design input parameters. Whereas the physiology part of the PBPK model, the flowchart that you have seen before, it's similar to that, but this is more comprehensive, which shows you that it uses quite a lot of parameters like blood flow to individual organs, what is the intrinsic clearance in those organs, what is the binding in those organs, those specific tissues, how the drug binds differentially. So in the background of a PBPK model, it's taking into account all these vast kind of scenarios that go on in your body. So once you put those two together, and with all the knowledge and the entire PBPK model you built, when you finally run a simulation, this is how it will look. So these dots here, it's a constant, it's a normal concentration time profile. So you have time on your x-axis, systemic concentration on the y-axis. So this is intravenous administration, this is oral administration. It's just on a log plot, so it's easier to see. So what happens is the clinical data is in the dots. So they come back and the clinical pharmacology team comes back to us and say, hey, you have the preclinical model. Now we have the first human data. Can you check if your model is predicting these so that we can have confidence in your model and trust it to do a prospective prediction, which means a prediction that you don't have data for future prediction, for understanding. So this is the confidence building part where we take the observed data, which you see for multiple individuals, which are the dots, and then we run a simulation using the various components that we discussed that go into a model. And the different lines, each line is an individual, which is the prediction. So we overlay with this and go, well, it seems to predict pretty decently for this model. So then we use, if it doesn't, if you're missing a portion, then we go about optimizing it so that we can recover this IV data because this is true data. So once your model predicts this data, you can go ahead and predict the oral administration. And like you can see, once it predicts this, it will predict the other doses and other routes of administration if your model is built robustly enough. So this is to show how when you put all of that together, ultimately how your model simulation looks like. I'll show you how you go from there to here, but this is kind of to give you a picture of what your final product is once you have built a model and how you use it against your observed data. So this will qualify as a pretty decent model because you have an equal distribution, and your model predicts the distribution and elimination pretty well, even the absorption. So it will be a good model to take forward into prospective predictions like drug-drug interactions and special populations and pediatrics. So with that, if I'm going to go into the next part and show you a demo of how this interface looks, how SimSip looks, and where are we inputting the data and um, how everything looks in the software. So I'm going to quickly switch screens here. Ma'am, can someone shout out if you can see the other part of the screen? Okay. All right. So I had to switch to a different server to be able to show you the SimSip. I'm going to just quickly check to see, can somebody confirm if you can see my SimSip screen or just um, type in so I know you guys are there. Think I'm still rolling. All right. So this is how SimSip look. This is how SimSip looks. It's a server, so we normally make reservations. There's different kinds of licenses, which is the technical end of things, but this is the platform. So when you want to use the PBPK model, um, this is the platform that we go to. So this is the human one, and I'm going to pull up the animal one as well. So let's do rat. Okay, so here we are. So the animal and the human modules are separate from each other. Like you can see, this is the animal module, and we have rat. We have quite a few species here that we can use to build a model. And where you'll build your um, this is where you'll build your compound. I'm going to focus on the human module, which is this part. So, like I said, we have various components to a model, right? So, this here is the population, which is where you can see there's quite a bit of populations that are already built and come with the simulator. So, we have healthy volunteers, cancer, Chinese population, cirrhosis, and there's different grades of cirrhosis depending on the disease state. We have geriatrics, Japanese population, pediatrics, pregnancy and preterm, renal and rheumatoid arthritis, and there's a dermal one too. So it's a quite comprehensive system. So what goes into all these is the various parameters that go into uh, building a population. So I'm not going to go into very much detail because it could be quite confusing. I just wanted to show you how everything looked based on what I have spoken so that you can kind of relate it to the real time modeling platform. So they have gathered so much data on the various organs, like how much cellularity is there. So you can see how comprehensive it is in the background. We have various enzymes in liver. They put in the skin, kidney, GI tract. GI tract is one of the important components where they built in your gastric emptying times, your residence times, what happens if you take food, what is the volume of your stomach, and the various parts of the intestinal tract altogether. So you also have tissue composition, tissue flow rates. These are all data that SIMSIP has gathered from various publications. They have given out grants to academic institutions to actually generate this data, as well as also from the clinical studies. They obtain this data to be able to build these populations. So, if you were to create a new population, if you have to create a completely new population for your application, if you cannot use one of these here, if you want to create a new one, you would need quite a bit of data to be able to build that. So they have brain, lung, there's quite a few. And if you are dealing with a cancer population, they also have a compartment that's your tumor compartment. So it specifies the size of your tumor, your blood flow, your blood volume, what's the volume of the tumor, and things like that. So that is the population part, which is the intrinsic part of what is within the body. So the second part is the drug parameters part. So we have population here, and then we have the substrate, which is the compound itself. So for the compound itself, these are all the drug intrinsic properties. And we need much lesser information on the drug compared to when you're building a population, because your body is a complex compartment. So these are physicochemical properties. So as you can see, the drug part of the model is also divided into various forms, various compartments. And I was saying the ADME part becomes the most important framework for this computational modeling. So we use molecular weight, log P, pKa's, and parameters like blood to plasma ratio and plasma protein binding. They are the most important, they form the backbone of the base model. Then, depending on the compound, we start building into the absorption model. So, if you have in vitro data, if you have heard about permeability, there's quite a few in vitro systems that you use to generate the permeability data. So you bring it all into here. And then there's the distribution part where you define your volume and your tissue distribution. So based on what amount of data we have, we can choose between a minimal or a full PBPK model and prediction methods. And that will determine how much drug is going to each of your tissues here based on their partition coefficients into those tissues. I might be throwing a lot of new terms out. I'm trying to keep it as simple as possible, but I will be happy to take any questions at the end of the presentation. So shout out if it's going into too much detail, but I'm going to try to keep it more simpler. The elimination part is where you define your clearance. So you have to define in a model how the drug is going out, right? So there's quite a few ways to do it. You have in vivo clearance, which means when you get the clearance from the clinical data, you take it and you plug this in. So if you have IV clearance, we use that. If you have oral clearance, we use that. And there's quite a few other ways of doing it mechanistically, which means you use certain enzymes and their intrinsic clearance, or you use organs like liver. If your compound is primarily driven out of the body by liver, you can actually define that in the model. And the comprehensiveness of the model depends on your application. What do you want to do with your model will determine how complex your model needs to be or how simple it needs to be. It really drives the application part, is what really drives the model building. What is the question that you're trying to answer? If a simple model can answer your question, then simple model it is. It doesn't always have to be a complex model that nobody understands, but it could be a simple model too. So it really is driven by the program team as to based on the questions that they are asking us, and then we determine what is the best way and what is the best kind of model that you can build. So when we are doing drug-drug interactions, basically there's another option here, which is grayed out right now, but when you build the actual model, you will be able to do it. Is So this is inhibitor. So as you can see, it defines it, inhibitor and substrate. So when you have drug-drug interactions, if you have two compounds, one becomes the substrate, which is the victim, and the other becomes the perpetrator, which is the inhibitor. So for a modeling like that, we can actually activate the inhibitor and then use one of the inbuilt inhibitors, or you can also build your inhibitor similar to how you would build your substrate here. And then we run a simulation. So basically, let me choose something that's already there. So I'm not breaking the confidentiality agreement with GSK. Um, so for example, there's midazolam. Sorry, there's midazolam. So I'm just the third part, which right, I forgot about this part. The third part is the trial design. Like I showed you, there were three components in the systems parameters, right? So this part is where you define your subject. So this is where you're defining, well, I need 10 subjects in each trial and I need 10 trials. So your population size becomes 100, and you can define the age, the target age, and the proportion of females to males. And then you have to define your dosing. So what is your dose of your compound and what is your route of administration? And then once you define everything in the system, you just run a simulation. So usually it can take um, it can actually take anywhere from 5 seconds to run a simulation to 24 hours to run a simulation. There were instances where we run overnight simulations. And here in the outputs is where you define what outputs you want from the model. For example, do you need liver concentrations? Do you need plasma concentrations? Do you need the AUC? So this is all the pharmacokinetics of the compound that you get. So demographic data. So there's general data, which means the patient related data. If you want to know what is the age, what is the size, and what are the characteristics of those 100 individuals that you're creating. That is all usually given in your demographic data. So when you choose your outputs and you run a simulation, it'll what happens is it gives out your results in the form of an Excel, and it's a huge Excel sheet, and we normally use what we need from this because it throws out quite a comprehensive Excel sheet. So like I was saying, this is your summary sheet. I'm just kind of getting you guys familiarized with how the model outputs are. They would be different for different platforms. For SimSip, this is how it is. It's quite a bit complicated, but not if you start understanding what goes into it. So as you can see, the demographic data is telling you how many individuals. So you asked for 100 individuals, right? So you have 100 individuals here. So for each individual, I don't know if you can see each individual, it gives you the parameters, the physiologic parameters for those individuals that they created. So the model went in the background and it created, let's 100 separate individuals. It's not the same individual, it's creating a population. So if you were to recruit 100 people, they would be different from each other, and that is what SimSip actually creates. It goes ahead and creates 100 individuals, like you can see here, 100 individuals, and each of them are different. Their age is different, their weight is different, their height is different. So it's basically you have data from 100 people without actually going into the clinic. And this is how it gives out. So if you remember the other um, output that I have shown you, that is, this is where it is coming from. So you have a concentration time profile. This is the mean profile, but you can also generate 100 different profiles based on your output parameters. So I think I'm going to not go into further detail on this because it could get confusing. But if I get any specific questions based on the interface, in the end I can go into it. But this is overall SimSip, and there's some really nice resources if someone is interested in SimSip. It's everywhere now. It's in India. They've been using it in the pharma companies in India, Novartis, BMS. Most of the, I think, medium to large pharma who can get their hands on these licenses have these licenses and are using them very much right now. So if it is something that you're interested in, there's quite a few resources that you can go into. And there was one another. Um, I also wanted to show. I just wanted to show how GastroPlus looks. I cannot go into a lot of detail with GastroPlus. It's not the primary software that I use, but I just wanted to show how the interfaces are slightly different when you're using different computational model um, computational model softwares. So the back one is SimSip. Well, before I go into GastroPlus, let me just tell you a little bit more. So there's a lot of equations that go into building this model and how they are talking to each other, all the pharmacokinetic equations and everything. So there is a back end, there is a front end. So all of those equations and the code and everything are in the background. So you can't see it in the front where the user interface is. This is the user interface where a person can go and build a model. But if coding and computing is another interest to you, that is something that they do as well. Here in the background are the people who are writing code to make this work and to write those equations that actually give you these outputs ultimately in your Excel sheet and calculating everything. So to keep it more simple and user friendly, they always keep it in the background, but you can always go into the help resources. And for example, if you want to know what method one is, or something like calculation of tissue partition coefficients, for example. So it actually tells you all the equations that it was built on. So this is the background. All these equations are in the back end of the development. I am a front end user. So as a modeler, I build models and I do these evaluations for the applications. But there are modelers who do the back end of the modeling, which is actually writing these equations into the model. So there's different aspects of being in a modeling area. So, and these are the two main ones, like a front end one and a back end one. So if that is something of interest to you, um, there is a possibility to work in that space too. So that was for SimZip. For GastroPlus, this is how GastroPlus looks. It's slightly different, but the basic idea is the same thing. You have the compound, you have the various molecular weight, log P, log D here. There's quite a few of the other things that GastroPlus takes into consideration. Then there's the physiology part, where it's talking to each part of your gut and different tissues in the body. The pharmacokinetics part, where you define your clearance, metabolism, or any other organ clearance if you see that. And then we just run a simulation here. And GastroPlus actually doesn't throw out a new Excel sheet, it just shows it to you in the same platform, and it actually creates a similar curve. So we use both platforms most of the time. We take a model from SimSip, try to build it in GastroPlus, and see how it is performing across, because the way that you can apply these platforms individually. So overall, the background is the same. The concepts that we learned about PBPK are the same across the platforms. It's just how the interface is looking and how you use it to apply for what your question is. So I think with that, um, I am coming to the end of the presentation. I hope the demo was helpful. It wasn't meant to confuse you further. It was more like kind of putting A, B, C, D together to show how the platform is. Um, there's some good resources. This is one of them. And if you go into Certara, you can create an account, and they have quite a few webinars and e-learning exercises there if you want to learn more about SimSip. You might not have access to everything, but you would certainly would have access to quite a few beginner level information related webinars. But I'll be happy to leave my email with either Vijay Bhargavi ma'am or Sumakant ma'am in case you guys need to get in touch. Um, and with that, I think thank you for actually coming out and listening to the webinar. I hope it helped kind of bring you to a new place or new understanding in PBPK. Even if I have just familiarized you to the terms and what we do, I'll be happy with that. So, with that, I can take any questions that you may have.
Yeah, there are some questions, Jennifer. Is it audible to you?
Yes, yes, I can hear you.
Oh, yeah. So your session is very, very good, ma. Uh, yeah, it is.
I
We are very happy that our student is went to this level and doing.
thank you,
Uh,
ma'am.
yeah.
It's really impactful work. I know that when I was in bachelor's to begin with, I knew I liked pharmacokinetics, but as you go further and get more exposure is when I learned these. And I have to say that I'm in a really happy position. I lead the US based modeling team here in GSK, and my projects are constantly with FDA and EMA. So the work that I do directly impacts a clinical study or a protocol, or we are conversing with big regulatory agencies. So it's quite a great opportunity, and I'm really thankful that I'm able to do that. But this is something, if somebody is in their bachelor's and wondering where am I going, what am I going to do, there's a lot of opportunity, not just in modeling, but anywhere they would want to go. And I came from there to here. So if I can, I think anybody could. So,
Yeah. Yeah. So there are some few questions, Jennifer. Shall we move?
yes, sure.
Some questions from Principal Madam.
Mhm.
So how you will validate the model, SimSip
So
model?
the validation, so it's a very important step, the verification. If I go back to that slide, ma'am, the overlay of the observed data on the predicted data. So once you get the clinical data, we take the observed data and we overlay our predictions. So if it doesn't overlay, it means it's not a valid model because it's not. Sorry, I keep going back and forth. Okay, here. So this is our validation step. So before we have any of the clinical data, we don't have these dots. So we are just shooting in the dark and saying, okay, this is our model. We are building this, and these lines are your predictions. So when they ask us to verify the model for a future application, we need the clinical data. So once we get the clinical data, we overlay the predictions with the observed data. And this serves as a validation, which gives us confidence that using this model, we are actually capturing what is happening in the clinic. So this particular step, validation can only happen once we have a clinical proper human data. And there's different levels of validation. You can have DDI data, you can have different doses. So you should be able to predict all the doses. Only then is it considered a valid, verified model, if it makes sense. Yeah.
Thank you, Jennifer. One more question. Can you suggest model development at institution level with available open softwares?
So honestly speaking, I am not sure, but I can find out about it. I know there is quite a few open source for PK parameters, like NCAs and stuff that come with. There's PK Prediction Pro, that you can use with Excel. So if you have a concentration time profile and if you want to generate the PK parameters, you can. There are certain open source softwares, but for applications like this, which are very specific to PBPK, I think it is still in a commercial level. But for interested universities, though, I'm not sure if you have a modeling based program, but GastroPlus and SimSip always give out grants to academic institutions, and that's how we wrote for a grant when I was in my PhD, from my school. And they give out like a 3-year grant. They will sponsor students, and they will also give like 7 to 8 licenses for that particular college, and they can use it. But the way to get that is like somebody from the faculty or from the institution needs to write a grant to SimSip or Simulations Plus and say, we want to do this, so can we get a grant or something like that. But honestly, I am not very familiar with this kind of modeling that is open source.
Okay. Okay,
it
Jennifer.
Yeah, I think it's quite expensive at this point, and they are not yet there to make it open source. They charge about $2 to $3 million for one license for 1 year right now. So it's quite expensive, unfortunately.
Okay. 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 One more question from Padmaja Madam.
Mhm.
Can we build PBPK model for biologics used in cancer treatment?
Yes, there is actually a special module for that, and it's really upcoming. They have put quite some work into that. They are looking at biologics, MABs, and in cancer populations, and also various HPV antivirals. In SimSip, I'm not sure there is one in Simulations Plus, but in SimSip there is a biologics module. Yes.
So these are the questions, Jennifer.
Sure.
Now,
Uh,
yeah, thank you for even though there is a lot of time difference,
no.
but you
The
accepted
sun
our
is up now.
invitation.
Yeah.
Yeah, yeah, you accepted our invitation and you delivered your lecture. Thank you.
Thank you so much for having me, ma'am. It was really nice to come, refreshing to come out of the um, my space and kind of go back to the basics and talk to everyone. So thank you for finding me and bringing me here to give the talk. I'll stay in touch with you or Sumakant, ma'am. Then
Okay, thank you, ma'am.
thank you.
Now I request Mrs. Anushree, Department of Pharmaceutical Chemistry, Assistant Professor, to deliver the vote of thanks.
Thank you, ma'am. A very good afternoon to everyone. So, we have reached to the end of this beautiful webinar series 2 on computational outlook in drug design, organized by IQAC in association with Department of Pharmaceutical Chemistry and alumni RBVRR Women's College of Pharmacy. I, Mrs. Hari Anushri, Assistant Professor, Department of Pharmaceutics, on behalf of RBVRR Women's College of Pharmacy, take the opportunity to propose vote of thanks to those who have directly or indirectly contributed to this webinar series. First, I thank Chief Patron Professor K. Mutyam Reddy Garu, Honorary Secretary Correspondent, for his support to us. I would like to thank wholeheartedly to our beloved Principal Ma'am Professor M. Sumakant Garu for providing encouragement and support for conducting this webinar. Thank you, ma'am. I take this opportunity to thank all the distinguished speakers in this series, Dr. Boyapati Sirisha Garu and Dr. Jennifer Mundunuru, for their excellent presentation and making this webinar very meaningful and interesting. Hearty congratulations to IQAC in association with Department of Pharmaceutical Chemistry and alumni coordinators Dr. M. Vijay Bhargavi and Mrs. G.R. Prasanna Lakshmi for such a beautiful webinar series. I thank registration committee Dr. K. Shruti, Mrs. Archana, Mrs. Hari Anushri, feedback committee Dr. B. Subbarao, Mrs. B. Padmaja, Mrs. B. Padma Bhavani, and all the teaching faculty of RBVRR Women's College of Pharmacy for their efforts in making this webinar series 2 successful. We would like to convey our sincere thanks to all the registered delegates and the one who are watching us on live for your active participation and sparing your valuable time on our sessions. We look forward for your enthusiastic participation in future in coming webinars. So here we have an announcement. Dear participants, the certificates will be sent to your registered mail IDs after filling the feedback form. The link for the feedback form will be available in the YouTube description box in our channel. Thank you. Thank you once again. RBVRR signing off. Thank you.
Thank you.